Welcome to Sigest, your fortnightly serving of digestible science from plant and food research.
Hello, I'm Blue Plunkett. Welcome back to our biosecurity and bioprotection series here on Sigest. In previous episodes, we have covered aspects of biosecurity and protection by discussing research on biosensors and host microbe interactions. Today, we have a specific case study about a disease affecting Kauri. To help us with this, we have an expert on the topic, Dr. Ian Horner, leader of the Pathogen Ecology and Control Team at Plant and Food Research. Ian, thanks for joining us. Before we get into your research, can you tell us a bit about your background and how you ended up in pathogen ecology and control?
Kia ora, Blue. I guess I started out life as a forest ecologist back in my university days and got a bit passionate about that, and then got hooked on plant pathology. And I've worked in that space for the last 30 odd years in horticultural science mostly, and looking at various pathogens and disease control and management and orchard systems mostly. And it's only more recently that I've sort of come back to my roots in native forest and Kauri forest in particular.
Right, so that leads into our topic for today, where the Kauri forests are in a bit of trouble. So what is this disease and how did it get here?
Good question. The disease, it's caused by this pathogen called Phytophthora agathodicida, which is a bit of a mouthful. But
quite a mouthful, yeah.
Yeah, it is. So the Phytophtheras are a group of organisms that have been known about for many years. There are well over 100 species and new ones being found all the time.
So is it a Greek word?
Phytophthora, yes, it is a Greek word. Phyto, plant, thora, destroyer or killer.
Right, so it sounds pretty bad already.
So it basically means plant destroyer. And as a genus, yes, they cause a lot of diseases worldwide. Probably one of the most famous would be the Irish potato famine, which was caused by Phytophthora infestans back in the 1800s. There's a whole heap of different diseases worldwide that are caused by these related organisms.
Right, and do we know how it reached New Zealand? Was it originally misidentified as an existing Phytophthora?
What we are now calling Phytophthora agathodicida was found back in the 1970s in one patch of Kauri forest on Great Barrier Island. And at that stage it was given another name. It was called Phytophthora hevii, which was, in hindsight, was probably a misidentification. But at that time, that was probably the closest of existing species that it could be described as, because that is quite closely related. Right. It's only more recently that we've worked out that this is actually a new species and hasn't been found anywhere else that we are aware of.
So to most people, it looks like a fungus or a fungi.
Yes.
But that's not actually correct. I was wondering if you could explain that a little bit.
Phytophthora, as you say, it does look like a fungus. It grows a hyphae, mycelium on an agar plate. It has spores. And for all intents and purposes, it looks and operates like a fungus, but taxonomically, it's not. It's not even closely related to fungi. It's a case of convergent evolution, I guess.
Right.
It's more closely related to algae.
Yeah, that's quite amazing. I tried to learn a little bit about it myself because I believe fungi have chitinous cell walls,
that's
whereas
right, yeah.
the Phytophthora are cellulosic, so that makes them more like...
They are more like plants than fungi.
Right. And so where is it found? How is it airborne or waterborne or...
This particular one is soil-borne. You won't find an aerial phase of it anywhere. It lives in the soil or survives in the soil and on plant roots.
Right, so is it? How is it detected? How do you find if a patch of soil is containing this pathogen?
In most cases, your first hint is that the kauri trees may be showing some sort of symptom. So that'll give you a hint that it might be there. And then what will happen is we would do a soil sample, and then that gets taken to the lab and tested. We have about a 2 to 3 week process that the soil goes through to extract the pathogen out at the end to give a positive identification.
Right. So when you're testing for it in the lab, is that like a DNA-based approach, or do you get it to move to a particular thing that it's attracted to? Or?
Yeah. So we've got a baiting system, basically. So you start with your soil sample, and we go through various steps of drying it and re-moistening it, and then flooding it. And this is all to try and stimulate the spore germination, so that it'll have dormant spores that are living inside the root fragments or perhaps even free in the soil. And we go through this wetting and drying process that stimulates these spores to germinate. And then it produces a little zoospore, which, think of it, this is a spore with 2 flagella. So it's a bit like a sperm with 2 flagella. And when you flood the soil, these swim and will swim up through the soil, and we float baits of a bait plant species on the surface. And the Phytophthora swims into that, infects it, and then we can put that onto an agar plate and get a positive identification. So it's a multi-step process, but reasonably straightforward.
Right. Yeah, that's quite amazing, because you don't normally, or most people probably don't normally think as water mold or this fungus-like organism as having a swimming phase.
Yeah, that's right. So yeah, it's got this little animal stage, and that is called a zoospore. That particular spore,
right? And so once it's close enough in the soil to a Kauri stand, what sort of happens once it encounters the roots of the tree? What's the infection process?
Yeah, okay, so these little zoospores, they will swim in probably only a few millimetres, perhaps a few centimetres through the soil. And then they will attack the Kauri feeder root. They insist on the outside of it, so they drop the flagella, and then they penetrate into that root and then start growing through the root with the mycelium that looks just like a fungus, effectively growing through the root and rotting the tissue as it goes.
Okay, right, yeah, I was going to ask, so, and then after this phase, you must start to see the symptoms start to arise in the tree? Or
well, you won't see the symptoms in the tree for a long time. I mean, it would, from, particularly a tree the size of a kauri, you could have a lot of feeder root infection before you saw any outward sign of anything wrong. And quite often, a tree might have been infected for many years, perhaps even decades, before you saw any above-ground symptom, particularly for the bigger trees.
Right, so that, I would imagine that makes the soil detection quite important if you're not going to see any outward symptom for many years.
Definitely.
Once the outward symptom does present, what does it look like on a tree? How can people see if a tree's infected or not?
Okay, so often the first thing people will notice will be a canopy thinning or dieback, hence the name Kauri Dieback. So you see the leaves will start to thin out, the whole canopy can look a bit sort of tufty and not as full as it should be. You can get a bit of leaf yellowing as well. So that's one of the symptoms you might see. The other symptom, and sometimes this will come first, is the lesion that might start to be observed at the base of the tree. So you get these bleeding cankers around the soil line and then advancing slowly up the trunk.
Right, so is this the girdling of the trunk that I've heard of? Yeah,
so eventually these lesions will spread and they'll work around the tree, so the tree eventually gets girdled or ring-barked and then dies.
Okay, wow. So if we take Waitakere as an example, the Waitakere Ranges, what's the level that this has spread to so far? Is it in half the trees, or you've seen it in a few of the trees so far?
Yeah, well, in the Waitakeres, there's been some quite good survey work done there, so they've got a reasonably good idea now of where the pathogen is. And it is widespread. There's a report that the Auckland Council have put out recently. Off the top of my head, I can't remember the precise numbers on infections, but it does seem to be spreading. It's certainly not in half the trees at this stage. I think, from memory, it's around 10 or 11%. It may be higher than that, I'm not sure. But it is certainly in a substantial number of trees. It tends to be in patches, so there'll be pockets of infection, and it's spreading from those pockets. But then those pockets of infection are scattered all through the bush. So,
okay.
Yeah.
And that kind of leads into the project that you've been spearheading, I believe. Is it the Kauri Research Project? Is that the title?
The Kauri Dieback program, which is the overarching program that's sort of a multi-agency team that are looking at that, and they are funding some of our research. So we're getting funded to do, in particular at this stage, phosphite research, looking at treatment of the trees. But they're also supporting other stuff, so looking at surveillance and where the pathogen is, where it isn't, that sort of thing.
Very cool. So how did you know to try phosphite as a potential treatment to this?
Well, phosphite's been used for decades now for treating various Phytophthora diseases. I first started playing with it back in the mid-1980s on apple trees. And it had dramatic results there in terms of healing Phytophthora lesions and cankers in trees. It's widely used in the avocado industry in New Zealand and worldwide. strawberry industries, a lot of horticultural industries use phosphite or various versions of it as a treatment for Phytophthora diseases. So it seemed a logical choice to have a go at for the Kauri forest.
Yeah, okay. I could imagine that the application method would have to be somewhat different for such, a large tree. How do you treat the trees?
Actually, the application method is very similar to what we might use in avocados, for example, which is trunk injection.
Oh, right, okay.
We have these specially designed syringes that you drill a small hole in the tree that's just sort of 6 millimetres diameter, drill a hole about 2 centimetres deep and inject the phosphite directly into the trunk. And that gets taken up by the tree, so it moves up in the transpiration flow, up into the leaves, and then gets redispersed right through the tree and back down into the roots.
So when did you first start this treatment?
We started with it just playing around in the glasshouse just to see if it was going to have any sort of efficacy against this disease. That was back in about 2010, 2011. We started our first field trials late 2011, early 2012, actually injecting diseased trees in the forest.
So that's 5 or 6 years now since you've started, and what's the results been so far? How's it looking for some of these trees?
The results have been pretty spectacular, to be quite honest. In terms of lesion healing, which has probably been our most useful measure so far, these bleeding cankers at the base of the trunks. We marked these wherever they occurred on trees and have been following the progress in response to treatment or non-treatment, so we had untreated controls for comparison. And what we found is that in most of the trees that we didn't treat, these lesions have continued to spread. So they just keep advancing and in many cases ended up girdling the trees and killing them. In every tree, and we treated about 120 trees in this trial, in every single case where those trees had lesions, those lesions stopped. They healed. That sort of wet, bleeding, sticky canker that you got just dried up, and in many cases started peeling back with healthy bark beneath.
Very amazing. That is impressive.
Yeah, it was very dramatic and quite exciting in terms of result.
Very cool. So now that you've got this result, what's the sort of next step for the project? Do you try and distribute this type of treatment to the public, or is there still some more fine-tuning to do?
Yep. There's still plenty of fine-tuning to do. The rates that we used in those early trials were probably too high, and we did get some toxicity effects within the trees. The trees have got a fairly low tolerance for the phosphite. So we've got, although it worked, although it cured the disease, or it stopped the disease spread within the tree at least, we did have some detrimental effects in terms of canopy thinning and a sort of almost a shock treatment in the trees. In some cases, not in every case, but in some. We've now dropped our rates considerably. Like we started with a 20% phosphite rate in the early trials. We're now using 4% in our more recent trials. So we've dropped the rate considerably. And the early results from that, which we're just sort of 18 months into that work now, are looking very encouraging. It looks like we're still getting that same sort of response in terms of lesion healing, even with a 4% rate.
Yeah, that's very good, because you don't get such a negative shock response,
right,
and you still induce whatever thing that's allowing the tree to heal.
Yep.
So do you think it's the phosphite directly killing the pathogen, or is it something in the tree that's responding to the phosphite?
It's a bit of both. Phosphite itself will have a detrimental effect on the Phytophthora growth. You can show that in agar plate tests and those sort of direct assays. But it is probably more importantly, it's stimulating the host defence systems. So it's actually stimulating the host to fight back against the pathogen. Now, the precise mechanisms of that are a bit fuzzy, but it's very obviously a host response as well as a direct attack on the pathogen. And you tend to get healing in a whole lot of systems with Phytophthora plant interactions, much greater than you'd expect from the concentration of the product that you put in there. You can have a very low level of phosphite in the tree that will still stimulate this response and the self-healing that you get with the tree.
And like you say, we can figure out the mechanism later. What we're trying to do now is just save these trees.
Yeah, sure.
And so one question I had, is it just the Kauri that are affected by this Phytophthora, or do you think that there's potentially other species that it can infect?
I mean, at the moment, Kauri is the only tree that has been noticed in the forest that has been badly affected by this disease. We don't know what other species might be affected. I personally feel that it's quite likely that there'll be other hosts for this particular pathogen. It may not be obvious on them. It might just be nibbling away on the roots, or perhaps even living on the fallen leaves, or eking out an existence on other hosts. I'm almost sure there'll be other things that it can live on without necessarily causing obvious disease on. We just don't know yet how important that is in the bush, but it's very important that we find out, because that is going to have an impact on how you might manage this disease and how you might consider it in an ecological sense.
Yeah, exactly. So how can we manage it at this point? I believe is it humans doing most of the spreading at this stage?
Almost certainly. It's because it's soil-borne. It doesn't move far on its own. I mean, its natural spread would be 2 or 3 or 4 metres a year, unassisted, and then there'll be just growth on roots or sort of passive movement through the soil. But when you get a vector in there, and that vector might be a tramper walking through the bush and picking up a bit of soil on their boots. It might be a pig running through the forest or wallowing around a diseased tree and then going somewhere else and having another wallow. Anything that moves soil in a dieback area will almost certainly be moving the pathogen as well.
So how can people help with this? What can we do? There's wash stations set up at the track.
There are wash stations, and they help. Unfortunately, the compliance of people using those wash stations is very low. You get responses from people saying, "Oh, I'm not spreading it," or "These don't work," or "I haven't got time to do that." But those are the people who are actually spreading disease. Either it's not deliberately. I mean, it's very much a passive process, but by not complying with those wash stations and not perhaps understanding how important they are actually being vectors for this disease and causing deaths of kauri trees.
Right, yeah, so compliance on the track sounds like it would be a big help.
That's a big help, but compliance alone will not stop the spread, because if you're walking through a bit of bush and you walk through an infected area, even on a track that might be a bit muddy, you pick up mud, you keep on walking through the bush, you might end up walking through a clean area. No wash station. And there's still the opportunity for people to spread this pathogen on their boots, even within the system, even if they're doing everything right as they enter the forest and as they leave the forest. While they're walking through the forest, they could still be transporting this pathogen. So
right.
That's a real problem, and there's not an easy solution to that, other than stopping people walking through the forest. But there's no doubt that it's tied up with people. When you look at particularly in the Waitakare's, where the most detailed surveys have been done, the disease spread or the disease incidence follows the tracks. You can very clearly see, and probably, I think it's over 90% of cases, the disease is within 50 metres of a track.
Wow. So how about people who have kauri stands on their own private property? Is there some sort of system in place or something they can do to try and save their trees?
Yeah, well, at the moment, we've just started this program called Kauri Rescue, which is funded through the National Science Challenge. And this is basically a group of community people and scientists who are helping the public to participate in the final stages of research. So it's quite an exciting project where we've actually engaging private landowners to inject trees themselves under supervision and under guidance and very clear protocols, and then monitor the trees and find out what sort of rates of the phosphite are protecting the trees or may be detrimental, to try and fine-tune that final stage of the research, to try and accelerate that. And it also gives these individual landowners who have got kauri on their own properties, can see these trees dying around them. It gives them a bit of hope. There's actually something that they can do to alleviate the stress on these trees, to actually potentially save them from dying.
Yeah, that's great to be able to get that engagement, and also that must be good for your research to get extra data and extra results.
Oh, absolutely. I mean, it will definitely speed up the final rollout of this sort of treatment. much, or probably more rapidly than could be done under the formal sort of science-led research trials by having the community involved. And as well, it's got a very positive spin-off as well as that whole community engagement and getting more aware of Kauri Dieback and just getting a bit of a buzz in the community. So I can see some spin-offs in that area as well. And the public enthusiasm is quite impressive.
Yeah, that's great to hear. Another thing I wanted to ask you about was, I know in some of the crops, a lot of the crops we work in, that when something like this happens, resistance is searched for within the population. Do you know if there's any scope, or have you looked in this direction at all?
I think in the long term that is going to be the hopefully the salvation of kauri forest, is that there will be some resistance within current kauri germplasm that will eventually sort of rise to the surface. I mean, if humans weren't involved, these things happen all the time in nature. You will get a new pathogen evolve or get exposed to trees. that tree will either be eradicated, which is unusual, but it can sometimes happen, or the resistance trees will survive, they'll produce seed, they'll propagate, the others will die. So you end up over millennia with resistant populations. I don't think we can wait for that to happen with kauri. And there are efforts at the moment to try and screen some of the diversity that is currently within kauri to see if there is any resistance to Kauri Dieback.
Is there much diversity in kauri after it got to quite low numbers, I believe, in the top of the North Island?
Well, that's the problem. I mean, kauri now, its natural range is sort of from about Raglan north, line from Raglan through to Taonga is roughly the natural range for kauri. So it had a fairly limited natural range before human intervention, but particularly in the 1800s and early 1900s with the logging, I think we're down to about 1% of unlogged forest. I think maybe about 4% overall of sort of regen and stuff in terms of what was there before human intervention compared with what's there now. So we're down to a pretty small pool of genetics, if you like, in terms of the kauri diversity. How broad that diversity is, I don't really know. That's not my area of expertise. There's certainly some diversity there, but the key is now to find out whether there's enough diversity to give us some dieback resistance.
I'm imagining, looking forward to hearing in the news or from your work some stand of trees that died where one lived and wasn't infected at all.
That'll be very interesting. This work's being led by Scion and Landcare scientists who are actually looking in detail to see if they can find any sort of differences within germplasm on whether there are actually resistant trees there. And I think something that needs to happen is targeting trees that are surviving in dieback areas, because it's not obvious. You go into a stand, particularly one of these regenerating ricka stands, perhaps in the Waitakaris, that have got dieback, and you don't see obviously healthy trees in the middle of a dieback stand. There'll be some trees that survive a bit longer than others, and it might be worth targeting those for seed collection. But you don't see really healthy trees in amongst a badly diseased stand. At least I haven't observed it personally.
Right, okay. Hopefully we might get lucky in the future and find one like that. Another thing I was wondering about, when you mentioned the zoospores, how they can they swim towards a particular chemical, or they must be attracted to something in the root of the kauri tree. Is there any scope, do you think, to try and put them off the scent, so to speak, or provide some sort of repellent that overrides them being attracted to the roots? Or
interesting question, and a really tough one. Monica Girth down at Otago University is doing some research on that and looking at the chemotaxis of the Phytophthora agathodicida to see what is attracting it, and are there things that might inhibit that interaction. So there could be something that could come out of her work, but it's going to be very tough. Even if you do find something that is perhaps repellent, how do you then introduce that into the kauri system in a way that would be beneficial and sustainable? But it could be that there are other trees that might be producing a chemical that might interact with the natural system in some way that could give some sort of protection. But it's going to be a really tough one, but an interesting bit of work to try and find out what these signals are
Yeah, very cool. And hopefully with treatments like what you're doing, we can buy enough time to try and figure these type of things out.
Yeah, and I think that's a good way of looking at phosphite. It's buying a little bit of time. It's not a long-term solution. You can't keep on treating trees forever. You can't treat all the trees in the forest.
And because the disease is still there, right, you're just halting it.
It doesn't eradicate the pathogen. It will perhaps have a curative effect on that individual tree that you treat, but the pathogen will still be lurking around in the soil, perhaps dribbling away on other hosts and just eking out an existence. As soon as the phosphite level drops in a tree that's not giving you protection anymore, that tree could potentially get reinfected. We don't know how long that period is, but I suspect it's quite a few years of
So
protection.
you're at 5 or 6 years at the moment without a retreatment?
Yep, that main trial that was set up in 2012 and the assessment done in early 2017, 5 years later, we only found 2 treated trees that had active lesions on them. All of the others were still inactive. So that suggests that we may get 3, 4, 5 years of protection from a treatment.
Well, that's great. That's buying us plenty of time to work on some of these other things. Well, thank you very much for joining us and sharing your research, and good luck with the next stages of your project. And hopefully we can manage to meet again in maybe 6 years, and you'll be able to tell us you've got 12 years without any progression of the disease.
Great. Thanks, Blake.
Thank you.
Cheers.
And thanks to everyone for listening. Please join us next time when we meet with Dr. Lisa Jameson to learn about her research on disinfestation. And as always, please send any feedback or questions to sigest@plantandfood.co.nz.